Guys, thanks for coming along today. I'm Geethika. I'm a fourth year at Corpus, and today I'm going to be going over the specific immune response. As you guys probably know, it's quite broad on your syllabus. I think there's like 6 pages dedicated to it, so there's quite a bit to go through. I've kind of picked a few of the high yield topics that we'll do. Sorry, my slides don't seem to be changing. One sec. Sorry, can you see the second slide now?
I think we are still seeing the first one now.
Just in the first round. Okay, give me a second. Yep. Has that worked?
Nope, still the first one.
I'm really sorry, it just doesn't seem to be responding. My laptop.
So if you go to non-full screen, do you think that could work?
Okay, I'm just gonna try sharing again and hopefully it'll work this time. Sorry about that, guys. I'm really sorry, my laptop seems to be having a problem. Do you think you'd be able to share it from the slides I uploaded to the OSME drive?
Yeah, I can try share the slides.
Yeah,
So
that
if
would
you
be great.
wanna
Thank
get
you.
your laptop sorted. Sorry everyone, I hope this is gonna get sorted soon.
Yeah, that's great. I can see
Okay,
that.
is everybody seeing the slides now?
Yeah, okay, we've got a thumbs up.
Okay,
So
and are you seeing the second slide now?
yes, I can see the second slide.
Okay, perfect. That should
That's
work
great.
then.
Thank you so much. Okay, yeah. Sorry about that, guys. But yeah, today I'll be going over some of the high yield topics in specific immune response. We'll cover hematopoiesis and like selection of B and T cell repertoire. And so this is basically central tolerance and primary lymphoid organ function. And then we'll look at secondary lymphoid organ function and induction of the specific immune response. And so this will include the antigen recognition and presentation by antigen presenting cells, then T cell activation and polarization into T cell subsets, and then also B cell activation and the germinal center reaction, which gives rise to antibody diversity and specificity. And then we'll also finally look at hypersensitivity reactions. And at the end, hopefully we'll go over a few multiple choice questions and look at one essay and sort of how to approach structuring an essay. Yeah, if we can go to the next slide. Thanks. Okay. So there are two main primary lymphoid organs in humans. So you've got the bone marrow, which is where hematopoiesis occurs. And so that's basically where you have stem cells that divide and give rise to the different like blood cells in the blood. And then the bone marrow is also where B cells mature. And then you've got the thymus, which is where T cells mature. And so as you can see here in the bone marrow, you basically, you have two main regions, which are sort of shown by the yellow region and the pink region in the middle. So the yellow region is the osteoblastic niche, and that's basically where the hematopoietic stem cells reside. And when they're in the osteoblastic niche, they're protected from things like radiation or cytokine fluctuations in the blood and other factors that could result in mutation or like damage to the cells. And so they're in this protected environment so that there's like a low mutational rate and the stem cells can remain functional for as long as possible. And then when they receive signals to divide, and so this is dependent on like signaling molecules and morphogens in the bone marrow like microenvironment, they the hematopoietic stem cells divide and they like migrate towards the pink region here, which is the vascular niche. And that's where they undergo differentiation and commitment into the different blood cell lineages. So as you can see here, and as you probably learned in first year as well, so you've got the hematopoietic stem cells, which then differentiate into multipotent progenitor cells, or MPPs. And then depending on the signaling molecules that they encounter, they get committed either to the lymphoid lineage, which is the MLPs, or to the myeloid lineage, so that's the CMPs. And so today, when we're looking at the specific immune response, we're more interested in the lymphoid lineage, which is going to give rise to the B cells and T cells. And it also gives rise to NK cells, but I'm not going to focus on them too much today because they don't come up too much in exams. And I think in second year, there's not as much that you need to know about NK cells. But yeah, so once the cells are at the lymphoid progenitor stage in the vascular niche, then some of them remain in the bone marrow, and these mature into B cells, while some of them will then enter the peripheral bloodstream and be circulated around the body. And once they arrive at the thymus, then they're taken up into the thymus stroma and they mature into T cells. So within the bone marrow, when cells are committed to a B cell lineage, they undergo the process called antigen independent development. And so that's basically where they mature into like fully functional B cells that express a B cell receptor, which is like a cell surface bound antibody, and they're also capable of secreting like IgM antibodies, which is like the most basic form of antibody. But at this stage, they haven't encountered any like antigens or any pathogens. Um, yeah, and so to have diversity in the like antigen recognition and like the capacity of the antibodies to bind to different proteins, there's a process that the heavy and light chain locus of the B cell undergo, and that's called VDJ recombination. And so this is basically a process in which a part of the VDJ locus of the gene is excised from the DNA. And this occurs at random. And so then depending on like which part is excised and like how the two ends of the genes are repaired together, you then get a high variability of antigen binding capacity that's produced. And so important to note at this stage is that VDJ recombination is catalyzed by an enzyme called RAG1. And the heavy chain recombination occurs first, and then that's followed by light chain recombination. So obviously when this recombination occurs, then you can have like it's a totally random antibody that's produced. So there's a high chance that it's self-reactive or like it could bind to self antigens. And so obviously that's not ideal because then you'll have an autoimmune phenotype. So they're eliminated by the process of central tolerance. And so here basically you have stromal cells, which are found in the bone marrow, and they present self-antigens, which are found in the tissue fluid, to the naive but mature B cells. And so if the B cell receptor that's produced at random by VDJ recombination is self-reactive and binds to the self-antigens, then it undergoes apoptosis. And so that's sort of a form of negative selection. And for some B cells, like the if the strength of the binding isn't too high, but there is still some level of self-reactivity, then they can undergo light chain editing, which is basically where they undergo further VDJ recombination. And so the light chain locus, like again, has a different part excised and is repaired again. And the aim of that is basically to hopefully get an antigen that is no longer, get an antibody that is no longer self-reactive. Yeah. If we can go to the next slide, please. Sorry, can we go to the next slide? Can you guys hear me okay? Yes, okay, thanks. I see the thumbs up. Yeah, let me just message Shalika. Okay, but in the meantime, I'll start telling you guys about how T cells mature in the thymus. Okay. I'm so sorry about all the technical difficulties today. This was not expected. Let me try sharing again while Eliska returns. Just bear with me. Sorry about this. Okay, can you see this now? Yeah, okay, great. I'm just sharing it as a PDF because PowerPoint doesn't seem to be working. Yeah, but anyways, so for T cell maturation that occurs in the thymus, as I was saying, so you have the lymphoid progenitors that are produced in the bone marrow and then have entered the circulation from there and are basically just circulating around the body. And then they enter the thymus in like a highly vascularized region in the center, which is called the corticomedullary junction. And that basically divides the thymus into two parts. The outer part, which is the cortex, and that has cortical thymic epithelial cells that make up the stroma. And then you have the inner part, which is the medulla, and that has medullary thymic epithelial cells that make up the stroma. And so the circulating lymphoid progenitors enter the thymus at the junction between these two, and they start to migrate first towards the outside of the thymus, and so that's like basically here going towards the outer cortex. And so at this stage, since they're just common lymphoid progenitors, so they aren't yet expressing T cell markers such as like CD4 and CD8. So they're called double negative thymocytes. And they undergo different interactions with the cortical epithelial cells. So an example of this is Notch signaling, which then promotes these cells to undergo VDJ recombination as well. And so for thymocytes, they're basically undergoing VDJ recombination in the T cell receptor locuses. And so the T cell receptor has sort of two main parts. It's got the alpha chains and the beta chains. And so the beta chain locus undergoes recombination first, and then the alpha recombination follows that. And following completion of VDJ recombination, these cells then upregulate both CD4 and CD8, becoming double positive cells. And that it serves two purposes. It basically shows that they're now committed to the T cell lineage. And it also then signals to the other cells around that VDJ recombination is complete. But then, of course, the same thing as with the B cells after VDJ recombination, since it's a random process, like the cells could have some self-reactivity. And so they need to undergo a selection process to ensure that they won't induce an autoimmune phenotype. And also for T cells in particular, in addition to the negative selection, they also have to undergo a positive selection process because for them to be functional T cells, the T cell receptor needs to be able to recognize MHC molecules because the TCR-MHC interaction sort of underlies the function of T cells. So the cortical thymic epithelial cells, or CTECs for short, they are a special type of epithelial cell that express both MHC class 1 and MHC class 2. So the randomly generated TCR, which like it has a random structure because of VDJ recombination, that will either it'll bind to MHC1 or it'll bind to MHC2, or finally it might just not bind to either, right? Those are like the three outcomes that could happen. So if it binds to MHC1, then the cells get committed to CD8 lineage, and so they downregulate CD4 and become single positive CD8 cells. Whereas if they bind to MHC2, then they get committed to the CD4 helper lineage, and so they bind so MHC2 binding cells will then upregulate CD4 and downregulate CD8. And then finally, if the TCR does not recognize any of the MHC classes, then it dies by neglect. And so in this process, because binding of this the TCR to MHC is like conferring a survival signal, that's then called positive selection. And so once the cells are single positive, they then migrate to the medulla, where they undergo negative selection. And so this is to prevent autoreactivity. So basically the medullary thymic epithelial cells, or MTECs, they are also a specialized epithelial cell type, and they express a transcription factor, which is called AIR, which that stands for autoimmune regulator. And so AIR is basically a transcription factor that acts on many gene loci, and so it results in just like self antigen expression of like proteins from most different tissue types in the body. And so as a result, you basically get the entire genome being expressed by these cells. And so you get all the self-antigens that are possible being found in the tissue fluid here. And so these are presented to the developing thymocytes by dendritic cells that are resident in the thymus. And so here, depending on the strength of the interaction between the TCR and the self-peptide MHC complex, then the thymocyte will have different fates. So if there's strong self-reactivity, then the cell will undergo apoptosis. So that's a form of negative selection. And then if they're moderately self-reactive, they become a special T cell subtype called Treg cells, or like regulatory T cells. So these are important in the periphery because they prevent autoimmunity and like help with immunosuppression when T cells might be activated by self-antigens, if that makes sense. And yeah, just on a molecular level, it's useful to know that Tregs express a transcription factor called FOXP3. And we'll come onto it later, like there are a few clinical syndromes that are associated with FOXP3 deficiencies, and that you don't really need that for multiple choice, but if you're interested in writing essays or something on this, then that's useful to know about. And yeah, if the TCRs are not self-reactive or have like low self-avidity, then those cells survive and become effector T cells in the periphery. Okay. Just do any of you have any questions on that so far? Is that making sense? Okay, cool. Let's move on then. Yeah, so that was how the primary lymphoid organs are structured and how lymphocytes develop. Then in terms of secondary lymphoid organs, the main ones to know about are the spleen, the lymph nodes, and Peyer's patches, which are like mucosal associated like lymphoid structures in the small intestine. So just a brief note on the lymph node structure. It's divided into follicles, which then have like the cortex on the outside and the medulla on the inside. So as you can see in the cross section here, you've got the primary lymphoid follicles, which are mostly where B cells reside. And then surrounding that on the inside, you've got the paracortical area, which has mostly T cells. And then right on the inside at the medulla, you've got the medullary cords, which are where most of the macrophages and the plasma cells, so those are the like antibody producing cells, they reside here. And so these follicular structures are then surrounded by lymphoid sinuses. And so you have the afferent lymphatic vessels, which enter towards the cortical side, and then they sort of surround all of the follicles with lymphs. In the various sinuses, you have like the cortical sinuses on the outside, the medullary sinuses on the inside, and then the medullary sinus just collects all the lymph and like it exits the lymph node through the efferent lymphatic vessels. And yeah, in terms of the follicles, so you have primary follicles, which are just mostly B cells. And then you have secondary follicles, which are the more developed follicles. And so they have a structure in the center called the germinal center. And so then that's where you have the antigen-dependent B cell maturation, and you have the like B cell and T follicular helper cell interactions there, which we'll talk about later. Any questions so far? Okay, cool. Just feel free to like shout out if you have any questions or put it in the chat. Yeah. So now we'll look at the induction of the immune response. So there's basically two types of immune responses that you can get with the specific immune system. So one of them is like the short-lived, like sort of immediate response to pathogens. And so that is, as I said, that's short-lived. With that, you don't get any like memory cells. It's just like purely if you have B cells that are being stimulated by an antigen, then they will produce like the default antibodies. So that's like IgM. There's no class switching. The plasma cells produced through this will die like quite quickly. But that's sort of the initial response, while the stronger response is like being formed in the secondary lymphoid organs. And so this, like not all antigens can initiate that, but like one important one that comes up is bacterial polysaccharides, like LPS, that can initiate an IgM response quite quickly through directly binding to the B cell receptors and stimulating IgM. But yeah, as I said, for the stronger immune response, that's like a multi-step process and it involves multiple different immune cell types. So you have first the antigen presenting cells. And so these cells, well, quite usefully named antigen presenting cells, they recognize antigens and then present them to other immune cells. So for example, this is like dendritic cells, which have special receptors on their cell surface called pattern recognition receptors. So these are like toll-like receptors, knot-like receptors, which you probably would have learned about in the bacteriology lectures. But yeah, so these receptors are specific to recognizing pathogen-associated molecular patterns, or PAMPs. So this could be things like LPS, as I mentioned earlier, which like that would only be found on pathogens, or for example, like single-stranded DNA, which again is only pathogenic because that wouldn't be found in like normal human cells. And so once the pattern recognition receptors are activated, these cells will upregulate a receptor called CCR7. And so that helps them to migrate towards the paracortical region where the T cells are found in the lymph nodes, because cells in that region will like secrete CCR7. So yeah, so the dendritic cells basically undergo like chemotaxis to migrate to this area. And yeah, cells that are sorry, I think my slides got it the wrong way around actually, but basically for endogenous antigens, so like antigens that are intracellular, they would be presented on MHC1, because basically you want to activate the CD8 cells, which are then going to be cytotoxic and kill the cell that has the intracellular pathogen. And then exogenous antigens are phagocytosed by the antigen presenting cells and then presented on MHC2. And so then these activate the CD4 helper cells to mount the B cell response further down the line. Yeah, so once the antigen presenting cells have recognized and are presenting the antigen on their cell surface, they then will interact with the T cells in the paracortical zone. And this will then activate the T cells and stimulate them to differentiate into different subtypes to mount different immune responses. So activation of the T cells requires three signals, and all three signals have to occur together for the T cell to be activated. And so that's kind of an evolutionary feature, so that T cells don't get overactivated, or like if there are self-antigens that sort of are similar to pathogens, you don't want the T cell to just be activated by any like one signal. You need sort of all three to happen together, so that it just increases the probability of there being an actual infection rather than it being a hypersensitivity reaction. So the first signal is the TCR peptide MHC binding. And so that, like you can see here, well, I don't know if you can see my pointer, but basically on the left-hand side of the diagram, you can see the TCR and MHC2 like forming a immunological synapse. And so that immunological synapse basically just means where two immune cells are interacting with each other, and like they transiently bind through interaction between their cell surface receptors. So that's the first signal. And then the second signal is a co-stimulatory signal. And so most commonly that'll be CD28, which is a cell surface receptor on the T cell, binding to CD80, which is found in the antigen presenting cells. And yeah, I know it's a bit annoying with immunology. There's just loads of like letters and numbers, which I did find quite frustrating while learning, but it's one of those things that you just sort of have to memorize a few of them. And yeah, so CD28, CD80 is a co-stimulatory interaction that provides the second signal to the T cell. And then the third signal is the presence of cytokines in the microenvironment. And so you have different cytokines. So for example, like interleukin 12 or interleukin 4, TGF beta. So some of them are pro-inflammatory, some are anti-inflammatory, and like depending on the combination, you would get different T helper subtypes being induced when the T cell is activated. And in terms of like how it works with all the different signals and like how do the signals really get integrated, it's basically by phosphorylation of second messengers. And so when you have co-stimulation, you're like exponentially increasing the number of second messengers that are produced and like phosphorylation of second messengers intracellularly. And so then that sort of raises, like it makes the concentration of second messengers enough to activate transcription factors, which are then required for T cell activation. But if you don't get the combination of signals, then you will never pass that threshold. And yeah, so the main transcription factors that activated T cells will express are NF-kappa B, NFAT, and AP1. And so these basically just, they're transcription factors that will work on different cytokine locuses. So for example, interferon gamma, TNF alpha, IL-2, which these are all like pro-inflammatory cytokines. So the activated T cells will express those. And so in addition to co-stimulatory receptors, you also get co-inhibitory receptors. And so these basically serve to provide some negative feedback to the activation process, because once the T cell is activated, you don't want it to then continuously be like rebinding to the same like antigen presenting cells, and then like again be secreting more and more of the same pro-inflammatory cytokines. And so then this works through like competition of the inhibitory and stimulatory signals. And so activated T cells will upregulate the inhibitory receptors. And so then the inhibitory signals can outcompete the co-stimulatory signals, and you basically get negative feedback and regulation of the activation process. And so one example of these inhibitory receptors is called CTLA4. And so that binds to the same ligand that CD28 binds to. And so they both compete for binding with CD80. And when the CTLA4 binding like outcompetes CD28, then the T cell will like not continuously be stimulated. It'll just remain activated at the right level. And these pathways have like a lot of significance in clinical application, because a lot of immunotherapy drugs and like monoclonal antibody therapy, especially for cancer, they target the inhibitory, the co-inhibitory processes. So, for example, there's one called pembrolizumab, which is a monoclonal antibody that interferes with the PD-1 and PD-L1 binding, and so that it blocks the inhibitory signal. And so T cells that are being inhibited or like deactivated in the tumor microenvironment then no longer receive this inhibitory signal and have a greater chance of fighting the cancer cells. Are there any questions so far on T cells? Okay, cool. This is just a bit on the T helper subsets. So I'm not going to go into too much detail on this because a lot of it is just like what signaling molecules are involved in like transcription factors and stuff. So it's kind of just things that you have to memorize. It's not super conceptual. But I think the main take home is that depending on the type of pathogen or the type of infection, you'd need different T helpers. So for example, if you've got intracellular pathogens, then you'll get more of the T helper 1 type CD4 cells. Or if you've got extracellular bacteria and fungi, then you get T helper 17. T helper 2 is interesting. This isn't really a function of the cell, but sort of a thing that it does is that it mediates allergy and asthma. So a lot of the like anti-allergy medications, they would then try to dampen the T helper 2 response. And then you've got the T regulatory cells, which again provide negative feedback and are for peripheral tolerance. And yeah, the T follicular helper cells are basically found in the follicles in the germinal center, and they interact with B cells to help activate them and mount the correct antibody response. Yeah. So the third part of the specific immune response is B cell activation. And so, as I mentioned earlier, this can happen sort of short term in a T cell independent way, where just the antigen directly binds to the antibody receptor, and then you get IgM. Or you can have the T cell dependent pathway, which is basically aided by the T follicular helper cells. And so the antigen binds to the B cell on the B cell receptor, and it's then internalized, digested, and presented on MHC2. And then the T follicular helper cell, so that's a CD4 T cell that's been activated by the antigen presenting cell or dendritic cell, is then differentiated into the T follicular helper and like migrated to the germinal center. So that T follicular helper will then bind to the peptide MHC complex that is on the B cell and will basically select the B cell for clonal expansion. And so it basically selects B cells which will have antibodies that are effective against the correct antigen on the pathogen. And so the way that this works is basically in the germinal center, you'll have 2 regions, the dark zone and the light zone. So in the light zone, you've got the T follicular helper cells, which bind to the B cells, and they induce clonal expansion. or they basically like activate the B cells. And so the activated B cells then migrate to the dark zone. And in the dark zone, they undergo clonal expansion. So that's like cell division by mitosis. And so normally, as you guys probably know, like mitosis has quite high fidelity rates. Like you basically get like the rate of mutation is not very high, right? But activated B cells are special in that they express an enzyme called AID, which stands for activation-induced cytidine deaminase. And so that's basically an enzyme that introduces like a higher rate of mutation in the genome when the cells are dividing. And so you end up, this process is basically called somatic hypermutation because you're getting more mutation than you normally would when the cells are dividing. And that results in more like BCR and immunoglobulin variability. And so this process is all like the end goal of this is to get antibodies that are more selective and have higher affinity to the antigen that the B cell is specific for. So when you get mutations, some of them will be higher affinity mutations. And so then these cells are selected again by the T follicular helper cells. And then this reaction sort of continues in a circle. And then after like a few rounds of like affinity maturation, then the B cells like terminally differentiate and will be sent out into the circulation as either plasma cells, so they secrete antibodies, or as memory B cells, so they remain for mounting a second immune response. Or of course, because the mutations will be random again, so you could end up with lower affinity mutations, and so then these cells will not bind as well to the T follicular helper cells, so then they'll die by neglect. Or you might get cells that just don't even replace their BCR, or like they get mutations in other gene loci, so they like they're basically unable to survive and they just die within the dark zone itself. But basically overall through this process, you're getting B cells, which they're all reactive to the same antigen, but they have slightly different affinities, and you're selecting for the cells which have the highest affinity to that antigen. Any questions on this? Okay, I can see on the chat, it's a question, how does the T cell know that these new B cells have higher affinity? T cells don't express antigens, right? Yeah, so that's a good, so it's not necessarily that they know that it's higher affinity, it's more that the ones with higher affinity are going to be taking up more of the antigen and are going to be expressing more of it on their peptide MHC. And so it's just like the probability then of a T cell interacting with that B cell is higher because it's expressing more of the peptide MHC on its cell surface, if that makes sense. Did that answer the question? Okay, great. Yeah, I guess with all of this, when I use words like no and stuff, but obviously the cells are not like sentient, but it's just the probability of binding. So if there's higher affinity between two molecules, then you'll just get more interaction between them. But the cells don't actually like know what's going on. Yeah. And then just briefly on the structure of antibodies, I won't go into too much detail on this because I think you covered this in first year, and I'm running out of time because of the technical difficulties. But yeah, basically you have different like isotypes of antibodies, And so the different isotypes are basically differences in the like heavy chain in the Fc region. And so they have the same specificity to the antigen and the same like antigen binding site. But because of differences in the Fc region, then they can like bind to different effectors. So for example, IgE will be able to bind to the FCE receptors on like mast cells and basophils. And so then they interact with those cells. Or like IgA will it's like produced more in the Peyer's patches or in like small intestine associated cells, and so it like binds better to effectors there. Um, yeah, and so the differences in heavy chain are produced by a process called class switching, and so that it's kind of like how VDJ recombination happens for the variable region. Class switching happens for the FC region. And so depending on like the cytokines that are present, then different like regions will be excised and you get the different classes. Yeah, and the last thing that I wanted to go over was hypersensitivity reactions. So there's four main types. Sometimes people will talk about type 5, which it's basically a subset of type 2. Yeah, so you've got type 1, which is the IgE mediated hypersensitivity. And well, sorry, before I get into this, hypersensitivity reactions are basically where the immune response is like the immune system is responding to something that it shouldn't. So this includes autoimmunity, so where you have like self-reactivity or reacting to self-antigens, or you have allergies, so that's like reacting to things that are not pathogens, so like things like pollen or peanuts. Yeah, and so you've got type 1, which is IgE mediated, and so these basically you get a response to soluble antigens, so antigens in the like serum or tissue fluid, and it's like mediated by mast cells and basophils that are degranulating and releasing histamine. And so histamine will then that produces like itching, swelling, so things like anaphylaxis or asthma, hay fever, or like allergic rhinitis. Those are all type 1. And then you have type 2, which is the IgG mediated cytotoxic or like stimulatory type. So cytotoxic is basically where the IgG will bind to cells and then mark them for destruction, for example, by NK cells or phagocytes. And then, yeah, stimulatory, which is also called type 5, that's where like the IgG binds to a receptor on the cell. And so it causes some sort of intracellular signaling. So for example, in Graves' disease, you get antibodies to thyroid cells that bind to the cells and stimulate them to divide and like produce more T3 and T4. And then you have type 3, which is the immune complex deposition, basically. So that's things like lupus, where you have IgG being like it forms complexes with other complement proteins, and it gets deposited in tissues and causes destruction to their structure. And then the fourth type is like cell-mediated or delayed hypersensitivity. And so that's basically where you get, it can either be CD8 mediated, where the CD8 cells are cytotoxic and they're destroying like self cells. So for example, in like type 1 diabetes, where you have cytotoxicity towards the beta cells in the pancreas, or you can have it as CD4 cells, which are producing a cytokine storm when there are no pathogens. So that's like, for example, in graft rejection. But yeah, um, I do have some multiple choice on the next slide, but since I'm presenting it as a PDF, unfortunately, the answers are already on the slide. But we can go over them anyhow, and if you just act like you can't see the bottom or like don't read it before answering the question. But yeah, basically, yeah, I don't know if you have any questions about it. It's kind of pointless with the answers there, but do all of these make sense? Or yeah, we can move on to the essay instead of doing these. Okay, I think we can just move on to the essay. So this is a question that I picked up from a past paper. I think it was 2018, if you're interested in seeing the examiner's report. But anyway, so the question was, what are the consequences of T-cell receptor ligation in the thymus? And how does this contrast with the recognition of antigen in secondary lymphoid tissues? So, I mean, I know you can see on there what my thoughts are, but does anyone want to volunteer like how they would think of structuring it or like things that they would want to include in their essay? You just feel free to like unmute and say stuff. Um, okay, well, anyway, so my thoughts were that you talk about sort of what first talk about what happens in the thymus. So that was like you have T cell receptors binding to the cortical thymic epithelial, you have them binding to the medullary thymic epithelial. So these are all like different interactions which have different consequences. So I think the keywords in the question that you want to focus on are, one is consequences. So you're talking about not only what is the interaction, but like what is the downstream effects of this interaction, or like why is this interaction relevant? So for example, if you're talking about the T cell and cortical thymic epithelial interaction, you want to then say that this is relevant for positive selection, or like this is relevant for differentiation of the T cells, etc. And then I think the second keyword is contrast. So you want to be drawing a direct comparison between the process in the thymus and the process in secondary lymphoid tissues. So like if we're saying that in the thymus, it's important for ontogeny of the T cells, whatever, then you want to say in the secondary lymphoid tissue, it's more functional, or like basically you want to draw a direct comparison, if that makes sense. And yeah, in terms of like beyond sort of the core stuff that we've been talking about, they like to see some clinical and experimental stuff included in the evidence, in the essay. So some of the clinical syndromes that you can talk about that are related to the specific immune system is, for example, like FOXP3 mutation, which I was talking about earlier, that results in a syndrome which is called IPEX, which the full form is a bit of a mouthful, but it does actually just tell you all the symptoms of the disease. So it's called immune dysregulation polyendocrinopathy enteropathy X-linked syndrome. But basically that just means that it's an X-linked condition that results in polyendocrinopathy. So you get things like type 1 diabetes or like thyroid dysfunction. And then enteropathy. So that's like problems with the like gastrointestinal system. So you'll get like diarrhea or nausea, vomiting. Yeah, and then similarly, you have a condition called APECED or APOSED, which is a result of like a mutation in the AIR gene, which we said that was the one responsible for the promiscuous self-antigen expression in medullary thymic epithelial cells. And so when that's mutated, then you don't get the central tolerance of T cells, right? And so you won't have like you'll basically have a lot of autoimmune phenotype. And so again, these patients present with like polyendocrinopathy. So that's like diabetes and thyroid dysfunction, adrenal dysfunction. And then candidiasis, which is candida infections. So that's like fungal infections, which so normally, unless you're immunocompromised, you don't get fungal infections. So here, clearly, since it's an issue with the immune system, then they're unable to fight off even fungal infections. They get candida. And then ectodermal symptoms is where you'll get like skin rashes. And then finally, dystrophy. Dystrophy is basically muscle weakness, right? So they'll have musculoskeletal disorders. Yeah, and I'm aware we're running slightly over time. So of course, like, if you do need to leave, no worries. But yeah, just the final clinical syndrome that is relevant to the system is you can talk about hyper IgM syndrome. And so basically that's where you'd see normal or high levels of IgM in the serum, but characteristically there'll be a lack of any of the other classes. So you'll have low IgE, IgA, and IgG. And so this can be due to a variety of reasons, but the most common is where you have an X-linked mutation in the CD40 ligand gene. And so CD40 ligand interaction is one of the interactions between B cells and T follicular helper cells, which is important in the class switching activation. And so then when you don't get this signal, these cells don't undergo class switching, so they only secrete the most basic form of antigen, which is IgM. And so then obviously with hyper IgM, clinically, they'll be more susceptible to infections, like especially ones that require IgG to clear. So things like intracellular infections. And yeah, in terms of experimental evidence, one of the ones that's like relevant to this question is that I thought you could talk about how the different T cell subtypes were discovered initially. And so that there's sort of a like seminal experiment which was done by Mossman and colleagues, where basically they characterized the cytokines produced by different T helper cell populations. And that was the first time that they found there were some cells that were producing like interferon gamma and IL-12, whereas there were some others that were producing IL-4, IL-13. And yeah, that's where the distinction was made between Th1 and Th2 cells, which yeah, obviously now since then it's been quite a few decades and we've found quite a few more different subtypes, but yeah, this was sort of the first time that things were divided into more than just CD4 and CD8 T helper cells. But yeah, so thanks for listening and sorry about the technical issues at the start. I hope this was useful for you guys. And if you have any questions, do feel free to reach out to me over email, or I can stick around for a bit if you have anything you want to ask now. And yeah, if you could please just scan this QR code to fill out a feedback form, that would be super helpful, just so I can know what to improve on and what I can do better next time. And at the end of the feedback form, you can put in your email address so that then we can send you a copy of the slides in case that will be useful for you.
Thank you very much, everyone, for coming in. Thank you to Geetika. I was just gonna say, if you do give feedback, which would be amazing, please comment mainly on Geetika's talk and rather than the technical issues, which I'm very, very sorry about. But we are obviously aware that this is something that needs to get fixed. So we are definitely going to try do that, but it's going to be much more helpful to hear your feedback about the actual talk. And yeah, really sorry about the delays and thank you for coming.
Yeah, I can just see a question in the chat, which is the class switching mainly just from secondary lymphoid organs? Thanks. So yes, it is from like once the antigens have been encountered. So that's all outside of the primary lymphoid organs. You do have IgD expression in naive B cells as well, but I'd say mostly it's secondary lymphoid organs where you've got the cytokines and antigens. There's another one. Does positive selection also occur in B cells? No, not necessarily, because with antibodies, like they will always bind to some protein, if that makes sense. Whereas with the T cell receptor, you need the positive selection because they have to bind to an MHC class. But with the antibodies, there's no such need. Okay. I think I hope that answered your questions, and if that's all of them, then yeah, just feel free to drop me an email later. But I think we can end the session for now.